for the invitation to talk to the group. It's really nice opportunity to tell you a little bit about a project that's sort of been bubbling away in our lab for a little while. It's far from complete, so I'm not going to complete a story as Jan told you, but I'd still like to tell you what we found because we're really excited about it. So I'm actually not very happy with the title I gave, so I'm going to give a different title, which is really the nutshell of what I'm trying to get at, which is trying to look at whether we can use experience to correct aberrant neural connectivity, and if so, how? Because I'm sure for this audience, I don't really need to explain in any detail, but of course, neural connectivity is absolutely essential for brain function. Aberrant neural connectivity will be devastating. And neural connectivity broadly can be said to arise through a couple of main processes. So the early intrinsically guided processes, such as axonal guidance, and then later the sculpting of activity by activity-dependent processes. What we're trying to look at is if something goes wrong in that early phase in axonal guidance, can we use enhanced activity at later stages to correct that? And if so, to restore function to the circuit? And if we can do that, what's the mechanism? And if we can uncover that, it could have potential for application to a variety of neurodevelopmental disorders. However, at this early sort of exploratory stage of trying to discover these processes, we're not quite ready to look at direct models of disorders. And instead, we're looking at our model of choice, which is the mouse visual pathway. We like this model because it's a really stereotyped organization. It's very accessible. It's very easy to pick up changes, and it's very easy to correlate structure with function, at least to a first approximation. So the mouse, of course, is a small ground dwelling mammal that's relatively low on the food chain, if you're thinking about vertebrates. And it uses its visual system in quite a different way to the way primates use our visual system. For example, it's not using it necessarily to detect ripe berries of different colors or to discover a good-looking partner. Instead, it tends to use its olfactory systems and its somatosensory systems for its day-to-day, a lot of the day-to-day things such as finding food and navigating, finding mates and so on. Instead, its visual pathway is important to enable it to do things like detect danger. And in particular, since it's small and ground dwelling, a lot of its dangers are coming from above or it's part of its dorsal visual field. And the binocular part of the visual field, where there's overlap of the projections from the two eyes, is expanded in this dorsal region. And it's thought that this expansion of the dorsal region reflects the importance of the binocular visual pathway for the mouse, because that's where it's got maximum sensitivity to detect important features. And we'll be coming back to that in a moment. If we look structurally at the pathway, the binocular pathway, of course, arises because that's where you have overlap of the ipsilateral and the contralateral, the visual fields of both the ipsilateral eye and the contralateral eye. And so the binocular part here is narrow, directly in front of the animal, shown here in purple, but as I said, would expand dorsally. And then there's a much larger contralateral monocular visual field going beside and even behind the animal. The ipsilateral pathway, of course, stays on the same side and maps consistently into the dorsal part of the lateral geniculate nucleus, whereas the contralateral retina, which sees this more peripheral part, maps to the rest of the lateral geniculate nucleus. And an interaction of various axonal guidance molecules enables these circuits to form with a high degree of topographic accuracy. The LGN projections then in turn project onto the primary visual cortex. And again, there's a stereotyped and organized mapping. So there's a nice topographic representation of the visual field from peripheral out here in the central part of V1 over to the central visual field, which is the binocular region in the lateral part of V1. This organization, however, can go awry, and as I said, is dependent on the actions of a variety of axonal guidance molecules. And one of the ones we've been working on for a while now is called 10M3, which is a type 2 transmembrane glycoprotein, which has been shown to be important for a variety of processes during neural development. And one of its roles, which we have been which we discovered and are focusing on here, is its role in the mapping of the ipsilateral projection, specifically into the lateral geniculate nucleus. So unlike the wild type, you see where it's confined to the dorsal medial part of the thalamus, the ipsilateral projections are spread along the entire long axis of the LGN. As far as we can tell, the contralateral projections project more or less normally, some subtle variations perhaps due to this change in the ipses, but by and large, they're fairly normal, and the mapping between the thalamus and the cortex is also maintained. So as a function of this, and we've shown this more directly, you basically get cells in this medial part of V1, which normally would receive inputs exclusively from the contralateral peripheral visual field, also receiving inputs from this part, or the central part of the visual field. And this seems to lead to disruption, serious disruption in the function of the circuit. And when Sam Merlin was in my lab many years ago now, one of the first things we did was to have a look at this using what was known as the visual cliff test. And I'm just using this as a sort of a bouncing ball to where I'm going next. So what Sam did, or one of the things he did, was to put the knockout mice into a box that had a clear perspex space and one half had a striped pattern. And basically he looked to see, did the knockout mice behave as if they could see that striped pattern. And normally wild type mice show a strong preference to stay over the striped part of the box. They don't like being here and spend over 90% of their time, the wild types are shown in black here, whereas the knockout mice spent roughly 50-50. If we put them in the dark, both groups behave the same. But the key finding here, because it leads to where we're going, was this experiment here, where Sam asked if it's the disruption of vision in the 10M3 knockout mice is due to the mismatch of the ipsilateral and contralateral projections. If we turn off the mismatch projections, at least going to one hemisphere, by acutely blocking the inputs from one eye with the sodium channel blocker tetrodotoxin, can we restore function to the circuit? And he found that indeed we could. So the mice remarkably started behaving very, very similarly to the wild type mice with respect to how much time they spent on the patterned versus the clear area. The wild type mice actually showed a decrease in performance, which he showed was due to a decrease in the size of their visual field. But this finding here was sort of proof of principle that if we are able to turn off the miswired projections, we can indeed restore function to this miswired visual circuit. And that, just to put on a diagram what was done here, so this is the normal circuit I already showed you, and you just essentially blocked activity in the eye so that you now have the uninterrupted mapping of the contralateral visual field onto one cortical hemisphere, and that was sufficient to allow function to the circuit. But it raises the more general question then, is can we then use this as a model to discover if and how we can restore function to miswired circuits, and importantly, what the underlying mechanism might be? Because we've got this beautifully organized pathway where we can correlate these different aspects with the structure and the function, and we can manipulate it, and it's a well-characterized pathway. So that's basically what we set out to do more subsequently. And the way we've approached this is using environmental enrichment. And I know some people here are extremely familiar with environmental enrichment. For those of you, who are less familiar, basically it's superficially simple, but actually very deep change in the way the mice are living. We put them, there's different ways to do it. We basically put them into an enriched housing at different stages, and I'll tell you what those stages are later. The enriched housing is characterized as being a much bigger cage. It has a running wheel for voluntary exercise. It has tubes and tunnels. It has jingly balls. It has smelly balls. And also it's being a larger cage, there are more mice in there with it. So there's enhanced capacity for social interactions. This contrasts with standard housing, where there's still elements of all these things. We're not depriving the mice in any way. They're getting the normal basal level of enrichment required by animal ethics protocols, but it doesn't have all these enhanced opportunities. So we had a look then to see if putting the mice into this enriched environment, if it had any impact on the visual circuitry in the 10M3 knockout mice. And we had a look first, because one of the beauties, of course, is that correlation between structure and behavior. structure and behavior in the visual pathway that's easy to assess. And so we looked at whether that we could use a different visual test to the one I introduced before, which we actually prefer for a couple of reasons, in that it's more ethologically relevant, in that it's emulating the escape of mice from an aerial predator. And in addition, it's actually using the dorsal visual field of mice, which is the binocular region, which is of most interest in our 10M3 knockout mice. So essentially the mice are just placed into a brightly lit open field, and above them there's a visual, there's a monitor, and the mice are allowed to explore the environment, and there's a petri dish in the center, and that contains a sunflower seed to encourage them to explore into the center of the box. And when they reach the center of their box and sort of start poking their nose around over it, we launch a very simple stimulus in their dorsal visual field, which is just a rapidly expanding circle. And this has been shown by other groups, particularly Yilmaz and Meister, to trigger a very stereotyped escape response, because it's thought to emulate the approach of an aerial predator. So just to show you, so this is a very robust, reliable response in wild type mice. You see here, and it rapidly escapes. Sorry, I should have explained, you're looking from the bottom here, and this little structure here is the shelter. So they're escaping into the shelter within a very short amount of time, and it's very reliable and very robust in wild type mice. If we do this in standard knockout mice, however, they really don't seem to respond to the stimulus. And remarkably unconcerned, they continue to explore and to potter around the cage. Eventually, they kind of wander off, but it's not a robust or reliable response as in the wild type mice. If, however, we take our knockout mice and we enrich them from birth, we got quite a different picture, where now the mice, after the stimulus is triggered, they now fairly reliably escape. Not with the same level of rapidity as the wild type mice, but certainly a recovery of the overall critical features of the response. So this was really promising. It seemed like enrichment was genuinely mediating an effect in these mice. And James, an honours student a couple of years ago, looked at this. And so those results I just showed you, those exact videos were examples from when enrichment was commenced from birth. We also looked at enrichment at different stages. So this graph just plots the latency to escape for the different groups. And you can see the wild type mice here shown in pale blue. Standard wild type mice have a robust response, responding within a few seconds. Standard house knockout mice don't really respond with any degree of reliability, tend to have very, very extended times following the stimulus before they will reach the shelter. If we enrich the wild type mice, there's no real difference. But if we enrich the knockout mice, we then saw there was a significant decrease in the amount of time it took them to reach the shelter, and the response became much more reliable. This change, though, that was induced by environmental enrichment only occurred if we enriched the mice from birth. And I should point out, this was quite a long enrichment period. We were enriching for a period of 6 weeks. So enrichment started a day or two before birth and continued to 6 weeks of age when this testing was done. So in that group, we saw an improvement. But if we started the enrichment just 3 weeks later at weaning, when the mice are still very young and the visual system is still quite plastic and undergoing development, we saw that there was no change at all. The enriched from weaning mice didn't show an improvement in their response, and enrichment in adulthood also had no effect. If anything, they actually responded a little worse, but that seemed to be to somewhat different factors. So there was clearly an early time window of sensitivity when the circuit was able to respond to enrichment to improve the behavioural response to a visual stimulus. We had a look to see how this related to the anatomical changes, just to get a feel of where in the circuit it was occurring. And initially, to be honest, I thought it was likely to be happening up in the visual cortex. But what we actually found was that the change predominantly is occurring subcortically in the lateral geniculate nucleus, in the site of that primary source of the miswiring in the 10M3 knockouts that I outlined at the beginning of the talk. So this is the wild type where we've injected a fluorescent tracer into the contralateral eye, and those terminals are labeled in green, and the ipsilateral terminals are labeled here in magenta. And we see the characteristic presence in the dorsomedial thalamus in the wild-type mice, both in standard and enriched. In the standard house knockout mice, you see that long tail of the ipsilateral projections right down into the ventral part of the LGN, which is the ectopic wiring that seems to be the source of a lot of problems with the vision of the mice. But when we had enriched them from birth, you see there's a significant retraction. It's not gone all the way back to wild type, but it's a lot more similar to the wild type than the standard house knockout is. Again, this anatomical repair of the circuit correlated with when the enrichment was commenced. So if we commenced enrichment just 3 weeks later at weaning, or if we commenced it in adulthood, we got the same result, where there was no repair down here, and that correlated with no restoration of the visual behavioural response to the looming stimulus. So this graph just quantifies some of that. So here we're just quantifying the percentage of the ipsilateral label, and basically what we found is this change here was correlated with a significant decrease in the proportion of the terminals that a proportion of the LGN occupied by ipsilateral terminals in the enriched from birth knockouts compared to the standard housed knockouts. And there was no change if we started at weaning or again at adulthood. And we did a topographic analysis as well, showing that this change here was significant. But basically the point here is we seem to be enhancing the pruning of the miswired ipsilateral projections, which could lead us towards a potential mechanism. In order to get at mechanism though, we also need to know a little bit more about timing, because enriching from birth to 6 weeks in a mouse, of course, spans the whole of development from when they come out and are in the sort of very early stage before the cortical layers form, before axons finish growing into the LGN, all the way up through the period of cortical plasticity into young adulthood. So 6 weeks is huge. We needed to know a little bit more about when. We had some clues, of course, because 6 weeks started from birth had an effect, whereas 6 weeks started from weaning didn't. So probably something going on in that first 3 weeks is going to be important. And so we just then did another project. Another honours student had a look to see, well, if we just enrich for 3 weeks, can we get an effect on the behaviour and on the anatomy? And that's what Justin did here. So he enriched the mice enriched the mice for 3 weeks, and then he did behaviour, which I'm not showing, but did show an improvement. And he also looked anatomically. And again, he found there was a significant retraction of those most miswired or ectopic inputs into the ventrolateral part of the LGN in the enriched knockouts. Interestingly, it wasn't as substantial as the amount of retraction that we got after 6 weeks of enrichment. So that was actually on its own quite exciting, because it suggested then that this time around 3 to 3.5 weeks postnatal might actually be a really critical time window where we can come in and we may find the process, find the mechanism is actually in process. So it's a good time for us to look at. And everything I'm going to show you from now on is going to be based on this particular time point. So we've got our little time window. What might be our mechanism? Well, as you probably know, there's been a huge interest over the last 10 years or so in the roles of microglia. Traditionally known predominantly as the brain's immune cells, they've been shown to play really important roles in neuroplasticity, mediating plasticity both during development and in later life, and playing roles in also a number of disorders. So microglia were a good potential candidate. And in particular, they had also been already shown by Beth Stevens' lab to play an important role in pruning of ipsilateral and contralateral projections in the LGN during early development. So just as a bit of background then, as you may know, microglia sort of can occupy sort of two main sort of states. One is the ramified or surveillant or resting state, which is, and it's seen morphologically by the presence of a large number of ramified long processes, or they can also convert to amoeboid or phagocytic, and this is when they're actively phagocytosing inputs. and we can label them with IBA1 and with the lysosomal marker CD68. And the morphology changes, IBA1 is upregulated, the morphology changes, and CD68 all is increased, and they're all markers of having converted to this more active phagocytic state. And so an honours student then took the challenge then of going on and having a look. So we know the place, which is at the most ventral extent of the mismapped terminals. And we had a good idea about the potential time to see, can we see any evidence of enhanced levels of microglia activity being induced by environmental enrichment that might help to explain what is going on in the pathway. And I hope you can see, these are just some qualitative images that we got very excited about early on, where you can see high levels of this blue stain, which is the IBA1, just here at the ventral extent of the label in the enriched knockouts, but it's absent from the standard knockouts. And when she looked at the lysosomal marker, CD68, again, we have high levels here, exactly in that position, which is much lower in the standard mice. And at high power, you can see there's a huge amount of upregulation of IBA1 and CD68, and these microglia look like they potentially could be doing the sort of process we were thinking about in terms of actively phagocytosing. Here's some more quantification to show that's really what is going on. So Lara quantified it in numerous ways, including the density of microglia, which is increased, suggesting they've migrated into the enriched mice to chomp away at those terminals. Changes in the morphology and the percent of CD68 expression, which is shown here in purple, is a significantly increased. And to quantify the morphology, she used this 3D solidity index, where she made a 3D envelope around the processes and compared the volume of the envelope to the volume of the microglial cell itself. And basically you expect it to be more thoroughly filled by the microglia when they've taken on the amoeboid morphology. And again, there was a significant increase, suggesting these changes in microglial morphology are consistent and are real. It doesn't actually prove they're phagocytosing the terminals. And so to look at that, she also did a phagocytosis assay here. So the CD68 is again shown in purple. Blue is just the IBA1 showing the microglia. And you can see these white blobs, which are actually inclusions in the lysosomes of the labeled afferent terminals, which are labeled from the retina. And there was a significant increase in the degree of retinal terminals in the lysosomes of the microglia in the enriched knockouts compared to the standard house knockouts. So she's been able to show that indeed environmental enrichment during this first few weeks is able to drive enhanced phagocytic activity of the microglia in a way that's actually able to restore wiring and restore function to the circuit. There's a lot more we want to do to follow this up. As I said, it's still at an early stage, and there's a number of experiments ongoing to follow this work up, but I'll leave it there for now. So just to conclude, so we're able to show that enrichment during just the first three postnatal weeks is able to drive corrective modelling of miswired retinal projections. And this is also associated with the recovery of visually mediated behavioural responses. And we've also shown this corrective model is being driven by an environmental enrichment induced activation of microglia. And we're quite excited about this because we think it's one of the first studies to show that enrichment is able to actually induce the microglia to remodel outside. And I should have mentioned, this is outside the time that the pruning is normally going on in the LGN. Normally that goes on in the first postnatal week. So it's outside that window. It's happening at around 3.5 weeks of age, and it's able to restore function to the circuit. And we think if we can get a handle more on this process and how we can regulate it, we can use it then to apply it to the treatment or development of therapies for neurodevelopmental disorders. So that's where we're hoping to go. Thanks for listening, and thanks also to a number of people who've contributed to my lab over many years. Atomu, of course, who's my partner in both science and life. Sam, Kelly, of course, although I didn't talk about Kelly's work, of course. Peter, James, Justin, Dylan, and Lara all contributed a lot to the work I talked about today. And the work initially started with Raganka, and Reinhard Fassler gave us the mice. And in the absence of any other money, we've been running this from crowdfunding. So I'd also really like to thank all the people who've supported our crowdfunding campaign so we can keep going. Thank you.
Thanks, Cathy. That was an awesome talk. I missed 10M3, and I can't believe the new behavioural data with the looming is. I mean, I thought the visual stuff was convincing, but the looming is pretty cool as well. So we have a question from Tony Hannon in the audience, and he says, "Thanks for showing your lovely and enriching work." He asks, as mice are mainly active during the dark cycle, do you think your environmental enrichment from birth may be acting indirectly, their effects on the mother, like through maternal care, lactation, or microbiota, and or non-visual parts of the brain and body of the pups or the juvenile mice?
I'd say almost certainly there's an impact from the mother. There's clear evidence that the way the mother interacts through the pups, it could be coming through the milk, it could be coming through touch. I think to address it more, we need even more information about the timing. I think the vision part's really important, and that's actually one of the things Lara's planning to. So Lara's gone on doing her PhD on this now, and it's one of the things she's planning to do in looking at dark rearing the mice to see if we need the window, to see how it correlates with eye opening. So yeah, that's one of the big questions we want to know is it vision driven, or is it other aspects of the enrichment, or possibly a combination? So yeah, great question, and we want to know that too.
And he had a follow-up question. Does the environmental enrichment of the wild type mice have the same effect on microglial structure or function, like including the phagocytosis and in the same brain regions?
Not at the time point we're looking at. So at the 3.5 week point, Lara's got all that data now. I didn't put it in here, but no, there's no effect on the microglia around the ipsicontra border in the wild type mice at 3.5 weeks. There could be an effect if we looked back at when the pruning normally occurs in wild type mice at around 5 to 7 days of enrichment, but we haven't looked at that time point because it's not where we're working.
Awesome. Thank you so much, Cathy. It's really lovely to see you as well. Hopefully the borders open soon and I can get back to Sydney to see everyone. But thank you for presenting your data, and yeah. Thanks,
Kelly.
And I'll pass over to Mike to introduce our next speaker. Thank you.